Thank you for the invitation. It's a pleasure to be here as part of this very interesting session. We're going to change topic slightly and go into the process that my lab's been studying for many years now, this process of vernalization, and how the mutants that we looked at to understand the molecular basis have led us into chromatin and chromatin dynamics. So my lab for a long time has been studying, what controls the timing of developmental transitions in plants. And the transition to flowering is the sort of archetypal transition that we've all studied, but the lessons from it, I think, have got important lessons for other developmental transitions and their regulation. And with flowering, it's clearly absolutely key for plants to get that timing right. You can see from the picture, if they get it wrong, reproductive success is pretty terrible. So there are many pathways the plants use to make sure that that transition to flowering occurs at the right time in favorable conditions to maximize pollination and seed set. Now, there have been a lot of labs over many, many years trying to understand, what are the sensors that actually follow these different environmental cues that are important. And the one we've studied is the requirement for prolonged cold. So many plants will flower only after winter, in that they need a very long period of cold, and then if they can only flower after the long cold, that they synchronize their flowering with the nice conditions of spring. And that's a process of vernalization, and that's the only one I'm going to describe today. So in a nutshell, and this is a lot of different people's work, the vernalization comes down to the regulation of one gene, a gene called FLC, flowering locus C. It's a Madsbox transcriptional regulator that basically transcriptionally suppresses a whole suite of genes required for the transition of the meristem to reproductive fate. So it's a transcriptional process, it's a break to flowering. And there are many pathways that then come along and switch that break off, prolonged cold being one of them. So vernalization or prolonged cold then switches off FLC expression, which allows flowering to occur. And that's an interesting process in that it's quantitative, and that's recapitulated on this northern here. With increasing weeks of cold, you get progressive reduction of expression of this MADS repressor. So one interesting question is, why does it take so long? Many cold processes in plants can occur much faster than this. And the second interesting question we wanted to address is, why is it then stable? If you take the plants out of the cold at the end of the spring, FLC expression stays very low for the rest of development and waits for the other environmental conditions required for the plant to induce flowering. So why is it quantitative? Why is it so slow? And then what's the basis of the epigenetic memory of that prolonged cold? And those were the questions we tried to address using a classic genetic approach, looking for mutants which disrupted vernalization somehow, and then trying to use those mutants to unpick the genetic basis, the molecular basis of vernalization. So this is what we did, starting from a vernalization requiring type, did mutagenesis, gave vernalization, and looked for the ones that that flowered a bit later than the rest of the plants in that experiment. Now, many years ago, the first mutant of the cloned gene that came out was this gene VERN2, which encoded a protein called SUSY12. And at the time, there wasn't very much known about SUSY12. There's now a lot known about it. It's part of the polycomb repressive complex 2 machinery, PRC2, well known in most higher organisms for chromatin silencing, for maintaining chromatin states. Now, this is the Arabidopsis protein. This is the Drosophila one as a lineup. You can look at the identity. It's, as I say, part of this PRC2 complex with 2 other major components. There's often other ancillary complexes, but there's a histone methyltransferase and another protein ESC. And what these do, this little protein complex, is it targets and methylates residues on histone tails, the lysine 27 trimethylation. So that is the signature of polycomb silencing. And the protein that we identified then as part of this complex that causes this histone methyltransferase activity. So at the time, that was quite a nice model. PRC2 was often, in many contexts, and at the time when we cloned this out, most was known about its role in Drosophila silencing, where PRC2 would act after other regulators and fix expression states. So here we had a nice model. Perhaps cold did something, and then PRC2 would act to fix that silent state and hence contribute to this epigenetic memory of this prolonged cold. Another set of mutants that came out were perhaps slightly harder to interpret. There were two mutants that came out of our initial screen, various names. Sorry, the names get rather confusing, but these two here, Vin3, Vern5, they were two of a little gene family in the Arabidopsis genome that were all structurally rather similar. They all encode PhD proteins. They also have a fibronectin 3 domain, and they have a very conserved C-terminus. Two of these came out of the mutant screen, the other two didn't, but as you'll see, some of those are also involved in vernalization. So we had PhD proteins which had actually always been associated with chromatin regulation because these PhD domains often bind histones or modified histones, and we had PRC2. How did all this fit together? Well, so far I haven't said anything about cold, except perhaps the PRC2 fixed the memory of cold. Well, one nice thing was actually the Vin3 expression was cold-induced. And actually it took a long time to induce it. It took up to 40 days for maximal induction. So that fitted with the dynamics of vernalization, and also the Vin3 expression decayed very quickly when you move the plants back into the warm. In contrast, the other protein of this little family that we got out of the genetic screen, so we knew was important for Vern5, was constitutively expressed. But this C-terminal region we found actually turns out to cause very efficient heterodimerization between these PhD proteins of this family. So when we look to see, well, do any of these function by working together or with any of the PRC2 complexes, these all homo and heterodimerize very efficiently through this C-terminus. Question was, how does that fit in then with the PRC2 complex? Now, the yeast 2 hybrid didn't show any direct interaction, but what was nice was when we went in and did affinity purification now with one of these PhD proteins, the constitutively expressed one, pull down the whole complex from plants that had been cold treated. What we found was all of these were in one complex. These had a Drosophila equivalent of all these various things, and if I draw them schematically, what we've got in this one complex, this is our bait, this is the PhD protein here. We have actually 3 of the PhD proteins, so one of the other ones that had come out of the genetic screen and another one. And all 3 of the PRC2 complex machinery, and then often there's this one here is an accessory protein. So we had our PRC2 complex and 3 of our PhD proteins as this cold-induced PhD PRC2 complex required for vernalization. Now, at the time, there wasn't an awful lot known about PRC2 complexes associating with PhD proteins, but actually those subsequently works come out that in both Drosophila and humans, there's a sort of modified PRC2, which also has PhD proteins. No homology, except for the fact they have proteins with PhD domains. So this PCL is the Drosophila protein that actually has 2 PhD domains. And what it does is, together with the PRC2 proteins, is superactivate that complex somehow. In vitro, it has much higher levels of histone metal transferase activity. So we think there's a functional analogy between these proteins that has evolved independently. Now, the other piece of data to put all of this together in terms of what they were doing on vernalization was when we looked at the dynamics of association of these proteins with our target locus during the various phases of vernalization. Now, here's our locus. Let me introduce it to you. There's FLC is the gene, remember. These are the exons. There's a very large first intron. It covers about 5 kb, this gene. Now, the first surprise was when we did the chromatin IP and looked at the polycomb complex, the PRC2, the Vern2 in our case. It was there already as the gene was active. So that was surprise number 1. Before the cold, the gene's very abundantly expressed. PRC2 is already there. That was different to what was sort of a canonical idea of polycomb silencing. What was happening during the cold was we had induction of one of our PhD proteins, the Vin3, and then assembly of this alternative PRC2, the PhD PRC2, and actually targeting to just one site in this intron. And during the cold, that's what happens. Just you get assembly of this PhD PRC2 in one site in the intron. and a small increase in the mark, the K27 trimethylation mark, that's induced by this complex. It was only when the plants were transferred back into the warm that you got actually a spreading of the complex along the length of the gene, now in the absence of one of the components, which of course is cold specific. And it was that spreading along the length of the gene that was associated with these very high levels of K27 trimethylation activity. And it's those very high levels that are required for the epigenetic stability through the rest of development. Now that, as I said, was slightly different to the picture in Drosophila. If you look at a textbook, talk about polycomb silencing, what you'll see is that certain regulators come along, switch the expression state of a gene, for example, various genes during embryogenesis in Drosophila, and then the PRC2, the core PRC2 comes along, causes K27 trimethylation, and fixes that expression state. So in the absence of the initial signal, that stays off. But actually, more recently, what's happened, certainly in a lot of the mammalian studies, is that what's come out is that there's a whole series of genes in what's called a poised or pre-programmed state. This is out of the embryonic stem cell analysis, where you have genes where you have both activating machinery, histone targeting machinery, histone methylation machinery, and also repressive machinery, so that everything's already there on the target, and it's just that the balance of their activities is what's affected. And somehow, either during differentiation you have, perhaps by modification, post-translational modification, or accessory proteins, you then shift the importance of one of the complexes over another. And certainly that seems to be the case for FLC. What we've got is the core PRC2 is already there. In fact, in the active state, there are histone methyltransferase complexes giving the other marks, the K4 trimethylation. There's all sorts of a range of marks associated with active transcription that are already there. And then you shift the balance of activities by now association of these little PhD proteins. So it seems to be a very conserved process in that we've got same kind of machinery, we've got a nucleation at a specific site, and then some kind of spreading mechanism, and that spreading mechanism gives very high levels of K27 across the whole gene, and that's what's required for epigenetic silencing. Now, there's lots of questions in the epigenetics field which we really still don't know very much about. Sort of the basis of, for example, why in any of these kind of mechanisms, epigenetic mechanisms, how can you have quantitativeness? What's the dynamics of this nucleation and spreading? Why does it only spread post-transfer to the warm? And that's true in many of the other systems. And probably one of the biggest questions in the epigenetic field are, are histone modifications themselves really the epigenetic marks? Can those be maintained in the absence of the initial signal? And so we've started to sort of use the advantages of vernalization in that you can separate the nucleation and the spreading, and you've got a nice long system. You give the plant a cold at a certain point of time, and then you have the rest of development to do your analysis to actually really unpick some of these basic questions. And the one I'm going to focus on today is the basis of this quantitative response that we see so nicely for vernalization. Now, as I've described to you, one of the early points of vernalization is that you have the targeting of this PhD PRC2 to one site in the intron of this target gene FLC. Now, so when we then go back and do a much more detailed analysis of what that complex does, which of course adds K27 trimethylation, through different phases of vernalization, we needed to set up a way of doing this chip analysis of K27 trimethylation in a very reproducible way, so we could compare quantitatively one sample with another. Far from trivial, let me assure you. But what Jie Song, a postdoc who's done all this work, managed to do was to generate the chip protocol so she could scan all the way along the gene, looking at K27 trimethylation in plants that have been given different treatments. So what you're looking at here are plants that have had no cold, 2 weeks cold, or 4 weeks cold, and immediately at the end of the cold, the chromatin's isolated and the chip's done. And what you can see is that there's not an even amount of K27 all the way along the gene, even before cold, but what I hope you can see is that what changes is the K27 just at this one site, this nucleation site where the PhD PRC2 associates during the cold, and that is quantitative. Low levels without cold, higher and higher with cold. When you then transfer the plants to the warm, and you get the spreading of this complex along the length of the gene, you then see the quantitativeness, if you like, spreading across the length of the gene. And again, 2 weeks is less than 4 weeks. So in one respect, we can understand the quantitativeness. You can see it manifested by the K27 levels. But what does it actually mean in terms of this quantitative behavior? Now, we were inspired at this point by a paper that came out some time ago now, modeling, just by theory, what happens in these epigenetic switches. So we've got a switch here from an active gene to a silent gene. This is a switch looking at mating type blockers in Pombi, where you have different histone marks. Doesn't really matter what they are. Histone methylation is the silent mark, histone acetylation the active mark. And what they were looking at was the modeling of the effect of how you can have a stable on and then switch to a stable off. Because the whole thing with epigenetic states is they're stable. But how can you then change something that's stable into a different state? And they proposed through, and at the time there wasn't much data to support this, but there is more practical data now, to say there's a positive feedback between the modification on the histone and the recruitment of the complexes that give that modification. So positive feedback. If you have methylation, for example, K27, you will recruit complexes that give you more methylation. And actually, the essence of this is that the methylation-demethylation is a very dynamic process. You know, we show these pictures of these, nucleosomes with sort of K27 stuck on them. And your picture is that it's a relatively stable thing. It's not stable at all. It's a very dynamic process. You constantly have to keep remethylating, demethylating, remethylating. And what they had shown was that you can actually have stability through this if you have, let's say, a K27 or a K4, in this case, methylation. You will recruit more complexes that give you more K4. And through that, you can be stable through cell division, because every day or so, the DNA will divide. You're going to have to regenerate that epigenetic mark. So you need to get stability, and then to be able to get switching, you need a dynamic, because if you're too stable, you'll never switch. And that dynamic is generated through this positive feedback. So you get more M if you have M, you get more A if you have A. You have to actually go through an unmodified state, and there's an element of cooperativity about all of these things, which we still don't really understand, which means that the various components come together, and then the outcome is more than the sum of their parts. That's an interesting session. But you also need long-range interactions to keep these, this silent state or this active state stable. So you have nucleosome turnover and a positive feedback recreating the mark, but because of that turnover, occasionally you'll switch to the opposing state. That was what was in this theory paper. So could we then utilize some concepts from that and actually try and understand vernalization? So the model was that, fairly simply, we had either an unmodified histone or a methylated or some other mark, an active mark, well, let's not say what it is at this point, an A mark. And you oscillate between those different states because of this turnover that's happening all the time. There is data now suggesting that, in fact, the K27 methyltransferase complex is recruited via the K27 mark. So there's a positive feedback. So there's some data backing this up. So what we have before the cold then is mainly a activating mark. Again, it's turning over constantly, but if you have enough of the A, you will recreate more A. So you have this turnover. After the cold, what you've got is mainly the K27 trimethylation, the M. And again, that's relatively stable because of this positive feedback. How do you then switch from one state to another? And the proposal is that we switch by actually causing this nucleation of this PhD PRC2 complex, which brings the machinery that gives you the K27 trimethylation to one patch within the gene. And as you bring that to the gene, you're then affecting the balance of the dynamics of these different complexes. And as long as your patch of M gets big enough, at some point you will then flip to the whole gene having this M mark all over it. Now, It's absolutely dependent on very rapid histone dynamics. The modelling assumes that nucleosomes turn over with about an hour, and perhaps the modifications in the same time frame. We've modelled cell division is perhaps 1 per day in the cold, 1 per day in the warm, and 1 per 4 days in the cold. So remember, you have to keep regenerating these marks every time there's cell division. But the switching is absolutely dependent on the fact we've got local nucleation of this opposite mark in one place, which then at some frequency causes a switch. Now, what do you get from that kind of modelling? What I've shown you is that we get increased levels of K27 through increased targeting of this PhD-PRC2 complex during the cold. And what we think that does is that that enhances the likelihood of then more, as you generate more K27 trimethylation complexes targeted to the locus, the likelihood of it switching over. You either then switch, and this occurs once you transfer the plants to the warm. You're either going to switch and, as I say, spread across the whole gene and then be stably a high K27 trimethylation state because of this positive feedback, or you're going to decay. And because this increases with the cold, what you're going to see is, in fact, and if I've drawn here a series of cells, so this is, let's say, our meristem, with mainly each cell showing active FLC. So and then you've got, because of the stability, you've got a few cells not expressing FLC. With increasing weeks of cold, you're going to get more cells in the inactive state, and after 6 weeks, you're going to have even more in the inactive state. So you have a bistable switch, and it's the population of cells that are changing, and that's giving you the quantitativeness. Now, is there any experimental data we've got to back that up? Well, in fact, why I drew them as blue is that we've now managed to make our FLC GUS transgene here, where before cold, most of the cells will express the GUS. If you give cold, and during the cold you see, and I'm not going to get into what's happening during the cold, that's another part of the lecture I haven't got time to go into, but FLC goes away, accumulation of the silencing occurs, so that if you give a very short cold and then you bring the plants back in the warm, the gene reactivates. And you see here, you are seeing reactivation in these patches. We think clones. What you've got here is genes where the silencing hasn't occurred and you've now reactivated. But in all of the cells which are white, the silencing, the spreading has happened stochastically, and you've now got epigenetic silencing. And if you give the plants long enough, then they all have this epigenetic silencing. So the other prediction that comes out of the model is that the slowness is actually what's happening during the cold, and that's something we've still got to tackle. Why is this so slow, this accumulation here? But the actual switching as to whether if you accumulate enough of this PhD-PRC2 at this local site, the actual switching to high levels of K27 across the body of the gene happens very rapidly. That could then be true for many systems, this rapid switch through of one state to another through a nucleation of an opposing modification. And in fact, when we look at that, the switching, and this is now the predicted timing of that switching versus the experimental data where we look at the K27 spread across the gene at different days after the cold, that fits very well with the modeling. So another prediction of the model is that the spreading is very rapid, and in fact, that's what we see. So we seem to have here an epigenetic switch caused by changed epigenetic states, but it's a bistable switch. You're either expressing the gene or you're not. And in fact, the quantitative nature then comes about as a result of an increased proportion of the cells which have switched. And in fact, this could then be a really very common mechanism for achieving a quantitative epigenetic response in many different epigenetic systems. Now, I just want to finish by very briefly talking about actually this in more real life. This is a sort of lab winter. You know, we put the plants in for 2, 4, or 6 weeks into the cold room, but what's happening there in the real world? And we've been working on natural variants which have very different requirements for vernalization. So here's some from Sweden, which actually need, instead of 4 or 6 weeks, they need more like 10 or 12 weeks cold to give you the full epigenetic silencing. If you take a rapid responder and give it 4 weeks, then FLC expression is low. This is what you're looking at here. Autumn expression high, during the colds low, and then epigenetically silent. If you take one of these very northern Swedish ones and only give it 4 weeks, it's low in the cold, it's just not epigenetically stable. It reactivates post-cold. However, there's nothing wrong with the machinery for vernalization, because if you give those plants a longer period of cold, then the gene is epigenetically silent. So what is it that's quantitatively accumulating during the cold? Well, we followed that up and showed that, in fact, if you isolate the alleles from these different Arabidopsis types and put them into a common background, it's cis-element polymorphism that's determining the different lengths of cold required for that epigenetic silencing. And we've gone on now and said, well, when we then go back to our chip analysis, can we actually see what it is that's actually changing in the alleles that need 12 weeks versus 4 weeks cold, for example? If we look at our K27 profile along the gene, this is going back to the gene again here, we're just looking at the nucleation region here, and we look at the K27 enrichment. This is in plants that respond rapidly. After 4 weeks, you've got pretty much maximal K27 enrichment in our nucleation region. So these are accessions that will epigenetically silence after 4 weeks. However, if you take your northern Sweden one and give it 4 weeks, you're still not as high as you would be if you were a rapid responder. In fact, you need 9 weeks to actually get to the same level of silencing. And that is due to cis-element polymorphism, presumably through some differential interaction with this PHD-PRC2 complex, and that's what we're following up. And when we actually look at the polymorphisms, and this shows a lineup of the rapid responding type, the Columbia allele, and where the polymorphisms are in this northern Swedish allele, and then do a lot of mix and match experiments to determine which of the polymorphisms are important, cutting straight down to doing a lot of analysis of, lots of transgenic analysis, doing a quantitative analysis of that transgene. It turns out to be these 4 SNPs that are contributing that quantitative variation. And these are the SNPs in our nucleation region. So the nice thing now, of course, is that the natural variation sort of intersects absolutely with the mechanistic analysis. We can actually begin to use the SNPs and the changed dynamics of what they're conferring to unpick the dynamics of the nucleation, to understand a whole series of non-coding RNAs that I've not had time to talk about, and what their role is in nucleation, and then to really understand the determinants for this epigenetic switching. So that just leaves me to acknowledge the people who've done the work. Jia Song is the postdoc who's done all of the K27 trimethylation work, really very difficult in all of the stochastic silencing, in collaboration with some modelers, Andrew Angel, who's the postdoc, and Martin Howard, the PI at John Innes. It's a very good collaboration that we've had with them, and Vincent Coustam, who did the work on the natural variation I talked about. And these are our funders. And thank you for your attention.
That was very enjoyable, thank you. You raised a question in your talk which appealed to me, because it was a question an undergraduate came up and asked me after I presented some of your data in a lecture, in which he said, is histone modification genuinely an epigenetic mark? And so I wonder, what criteria would it have to pass to be an epigenetic mark? Because I was unable to answer the question. So.
Well, the criteria is that you have to have some mechanism for copying, like we heard with the DNA methylation. Somehow you have to, if you have a locus and through replication, you somehow copy the histones that were in the parental chromosome onto now both daughters, so that in the absence of any signal that's generated it in the first place, it can maintain itself through cell division, then you would describe it as epigenetic. There's a lot of debate in the field about this. People say, there's no obvious mechanism for that copying, therefore there must be some underlying protein feedback or something like this, which is then conferring the histone modification. So the actual epigenetics is the kind of underlying, if you like, complexes that are the ones that are managing to maintain the state, and the histone modifications are just an elaboration of that. I think the data that's coming out that says that there's good a feedback, so that if you have, and this is true for acetylation now, for K9 trimethylation, for a lot of the modifications, they tend to recruit the complexes that generated them. And that, if you like, gives you the basis for how you could imagine they are epigenetic. They don't need some other factor. But there's a very good review that Danny Mojed has just written in Cell that's just come out on the whole of this, is the feedback sufficient to explain histone modification, or do you need some kind of DNA, sort of DNA sequence interaction to actually give you the targeting? And he would argue actually you need a bit of DNA sequence, a bit of non-coding RNA, and this positive feedback, and together that cooperativity gives you sufficient oomph to actually be able to fully copy, because it has to be exceedingly robust. I mean, if the dynamics are as fast as people measure them and as predicted, you have to have a very good system to maintain this all the time.
So those 4 polymorphisms that you found when you did your transgene experiments, do you require all 4, or does each one quantitatively contribute? And also, can you go look at some of the other ecotypes now and predict their phenotypes based on?
That's right. You would have thought we'd have been able to go in, and the simple answer to that is that's a very rare allele. Those 4 occur in other accessions, but not in the same. You don't find another accession with the same 4. At the moment, we can actually divide that into 2, and the 5'2" and the 3'2" are actually sufficient to give a reactivation. So it's not just one polymorphism, it seems to be a context of the 4 together. And that accession has that 4 is a rather rare allele. But there's now a whole interesting story to look at the accessions and to actually understand the polymorphism, and then you get into cryptic polymorphism, such that you have polymorphism that's hidden until you have one more polymorphism, and then suddenly, you have a very big effect. So you get into some rather interesting population genetic analysis.